The Lethbridge High School iGEM team presents Dia-Beatable, a new siRNA-based treatment for type 1 diabetes.
iGEM, or International Genetically Engineered Machines, is a global competition. Teams from around the world spend a year developing a project using synthetic biology to solve both local and global problems in the world. The main event of iGEM is the Jamboree, which takes place in late October. During the Jamboree, hundreds of teams on the university and high school levels come together to showcase their projects to a panel of judges. Judges will give their advice for improvement and award medals to team projects. Teams will also each make a wiki for other teams to learn about their project.
The Lethbridge High School iGEM team is composed of 21 high school students in Lethbridge. The team is mentored by staff and undergraduates from the University of Lethbridge. The team meets once a week on Zoom to discuss general updates and plans for the future. However, most of our work happens off-meeting in smaller groups.
Type 1 diabetes, also known as diabetes mellitus, composes 5 to 10% of diabetes cases worldwide. Type 1 diabetes occurs when insulin-producing cells malfunction and do not produce enough insulin. Insulin is a hormone produced by the body whose purpose is to break down glucose. So, a lack of insulin causes a buildup of blood sugar. Type 1 diabetes symptoms include unexplained weight loss, intense thirst, hunger, and increased urination and fatigue.
Diabetes type 1 affects over 9 million people as of 2022. Furthermore, diabetes is the 9th leading cause of death in the world. Deaths related to diabetes type 1 are mostly due to cardiovascular disease and a lack of treatments in low-income countries, most prevalently in the African region. However, other deaths can be attributed to individuals refusing to follow treatments and daily recommendations due to the lengthy time commitment. Thus, type 1 diabetes is a current problem that needs to be addressed in order to reduce the lives lost and improve the daily lives of diabetics worldwide.
The main treatments for type 1 diabetes used today are keeping the disease under control through healthy eating and exercise, and regular insulin injections to increase glucose breakdown in the blood. Managing the disease through a healthier lifestyle can be difficult, and medications can have side effects. For many people, daily insulin injections are required, which are physically painful. There are also pancreas transplants that can eliminate the need for treatment. However, a problem with these transplants is the interruption to a diabetic's daily life and the high costs of treatment in some countries. For example, in Canada, a diabetic with type 1 diabetes must spend about $2,500 per year on treatments. A more extreme case is the United States, where diabetics must spend an average $12,000 a year.
The goal of our project is to create a glucose-dependent self-amplifying mRNA complex that codes for human insulin. This construct may contain a few parts to accomplish this, including a glucose riboswitch, sequences necessary for self-amplification, and the human insulin gene sequence. The self-amplifying mRNA attempts to mimic the natural processes that alpha viruses carry out to create substantial amounts of subgenomic RNA encoded to create viral structural proteins. Self-amplifying mRNA codes for non-structural proteins within its sense strand to create a complementary negative sense strand. This complementary strand is used as a template to produce copies of the original sequence, therefore amplifying the sequence to increase the quantity of the target mRNA and increasing the stability period of the mRNA in the cells. The sequence for human insulin was found in the National Library of Medicine database as the Homo sapien chromosome 11 wild-type insulin gene. While within the cell, insulin is expressed as a 110 amino acid long protein, our construct will only compose of the DNA sequences required for the mature, active form, which consists of only 51 amino acids, thus making post-translational processes unnecessary. Additionally, our construct also includes a copy of the human Kozak sequence used for protein translation. This sequence helps position the start codon into the active site of the ribosome and is necessary for protein synthesis in eukaryote cells. To make our construct responsible to blood glucose levels, we will develop a glucose riboswitch. We aim to create a glucose aptamer, whereupon binding glucose, expression of any downstream genes, including insulin, will be triggered. We first will test our riboswitch designs in vitro. Fusion of the riboswitch aptamer domain to a dye-binding fluorogenic aptamer will create an RNA-based fluorescence biosensor. The ligand, which is glucose, binds to the native riboswitch aptamer domain, which then allows the fluorescence aptamer, which will be our RNA mango, to bind to the profluorescence dye and activates the fluorescence. This will allow us to test multiple RNA sequences and fine-tune glucose binding before we test responses in vivo. Currently, we will also work to develop an insulin riboswitch, which instead of turning on transcription of the downstream gene, will bind to the target ligand, which is insulin, to the aptamer will stop transcription. mRNA expression will therefore be controlled by blood insulin levels as opposite to the blood glucose levels. This riboswitch will be tested similarly in vitro for accuracy and efficiency.
Researchers from the Department of Biotechnology at the National Institute of Pharmaceutical Education and Research in Nagpur, India, have developed an insulin RNA aptamer that we will utilize as part of our first steps to creating our own insulin biosensor. Similarly, a glucose-binding RNA aptamer was reported by Naoki Sugimoto's lab at Konan University in Japan in 1998. However, little work has been done on this aptamer since, but it will be a good starting point for our project. RNA Mango has been used extensively to label RNA for imaging, and its cousin Spinach 2 aptamer has been used to construct previous SAM sensing RNA-based fluorescent biosensors. Our team also has a previous experience using RNA Mango as it was integral to our 2019 iGEM project, CRISPR-assisted detection and removal, CADAR.
This summer, we propose to design a DNA template for our RNA riboswitch biosensors and order DNA construct from IDT, PCR amplify the DNA sequence of our riboswitch, use T7 RNA polymerase to in vitro transcribe the RNA riboswitch, and finally test multiple glucose and insulin concentrations for binding to respective RNA aptamers using fluorescent signal of RNA magno. In the future, our project will need to be tested in eukaryotic cells. Tests will include mRNA stability, glucose sensitivity, and insulin production levels and rates.
The Lethbridge High School iGEM team is planning to volunteer with Canadian Blood Services to learn more about the current help available to diabetics. Volunteering will allow us to engage with people who have diabetes type 1 to hear their thoughts on our project, Dia-Beatable. We will also be collaborating with the new University of Manitoba iGEM team and Dr. H.J. Wyden. Their team will be helping us with our lab work and with their expertise in RNA biology. We hope to also reach out to the Open Insulin Project to gather and incorporate their feedback into our project. They are a nonprofit organization that aims to use synthetic biology to make insulin and diabetes treatment more accessible.
Our team is involved in many aspects outside of wet lab work. Members of our team are currently organizing a podcast to inform the community about iGEM and our project. We are also planning to conduct research on marginalized scientists to bring awareness to all scientific research around the world. Being involved in the community is an important aspect to us, and we are contacting organizations for volunteer opportunities to connect with our community.
Thank you for watching this presentation. We, the Lethbridge High School iGEM team, and everyone else contributing to developing Dia-Beatable, hope you've learned something, and will support our project in the future to improve the lives of diabetics.